Just kidding. We actually have a presentation from Dr. McCorvie. It's a little too expensive to travel from the UK for a 10-minute presentation, but he's one of the researchers we have funded. So let's see if I can get this queued up.
Associate in the lab, Professor YHU, based here in the UK at Newcastle University. And today it is my pleasure to talk about our HCU network funded work, which is crystallographic-based fragment screening to develop pharmacological chaperones for classic homocystinuria. So every day your DNA is being transcribed into mRNA, which is then being used by your ribosomes to produce various proteins that function in your cells that do the everyday job to keep you alive. What the ribosome does is that it produces this polypeptide, which is a sequence of specific amino acids, which then spontaneously folds into the native protein, and the sequence of these amino acids dictates which fold and which protein is produced. So in a normal situation, these native proteins, they have the correct shape, they're soluble, and therefore they're active, they're able to do their function. But in cases of inherited disorders such as classical homocystinuria, there's a genetic mutation that usually results in a missense mutation, and that is one that changes the amino acid sequence in the polypeptide chain. What this can cause is incorrect folding, and it prevents the polypeptide from ever actually getting into the correct 3D structure that is necessary for its function. In some of the most severe cases, it can cause aggregated protein, and in some of the less severe cases, it can slightly misfold it. But ultimately, what occurs is that the enzyme has the incorrect shape, and therefore it's not able to carry out its function, so there's low activity or none. Now, what we want to do is find a small molecule that binds to the mutant version of CBS and then causes it to form into the right shape and therefore rescue its function. And it has been recognized that classic homocystinuria is a protein misfolding disorder in the literature, and many researchers have been looking into this as a possible treatment for classic homocystinuria. So what we wanted to do is take a different approach in terms of finding small molecules, small drug-like molecules that bind to CBS. And from this, we wanted to take advantage of our previous success, which is determining the crystal structure of human CBS, which we did in 2014. And this was using a construct of CBS, a CBS loop deletion, which deletes this region in the protein. And what we want to do is to produce loads of this recombinant protein to then produce hundreds and hundreds and hundreds of this CBS loop deletion protein as crystals. And within these crystals, the CBS protein, the CBS dimer, forms this ordered array, which then we can use to soak small molecules in. And then we take these soaked crystals with these small molecules to the synchrotron that produce X-rays to allow us to solve the structure of the protein within these crystals. And hopefully, in some circumstances, these small molecules bind to pockets or on the surface of the protein, and we should be able to determine their structure and then use that information of these identified hits to then develop them into drug-like molecules. So we've already attempted this with CBS loop deletion, and here I show some example of the crystals that we've attained. They usually form these long rod-like shapes. And what we find within these crystals is that the CBS molecules pack in these order arrays where they actually have these large solvent channels. So these are large gaps that contain water, which are actually ideal for allowing small molecules to be soaked in and hopefully bind to the surface and on the pockets of CBS. However, we did find that we had a lot of issues in terms of the data quality that we were getting from these crystals. And this is judged in terms of resolution. And what we're finding is that most of the CBS crystals really only gave data that was in the lower end of resolution. So this is around 3.5 angstrom resolution. You can see that this distribution plot is skewed to that. And what we really want is we want it more skewed to the left. So the lower the number, the higher quality of the data, the more confident we can be with the findings that we get from these crystals. So we did try a brute force approach. We've soaked about 1,000 crystals. We've collected many data sets, but unfortunately, we just didn't get enough information from these crystals to be confident with any of the possible hits that we could find. So we've been trying to optimize another system, or two systems in this case, that could be used for soaking of crystals. Now, in one instance, we're still working on the CBS loop deletion, and what we were finding with this protein is that the enzyme was degrading, but with some modification of the genetic sequence of this construct, we were able to reduce this degradation. Additionally, we have been looking into another construct of CBS, which is just the regulatory domain, which we also crystallized in 2014. And at the moment, both these constructs are behaving quite well. They do give us crystals that are far better than the previous construct and give more consistent data that are more ideal for fragment soaking. And these crystals are now being optimized for future fragment soaking campaigns in September this year. I also want to mention some of our previous work on CBS that has been recently published. So it hadn't been known what the full-length structure of human CBS was. And here I have taken advantage of the advancements in cryo-electron microscopy. So in electron microscopy, we're just looking directly at the protein. We don't need to form crystals. And here I'm showing an image, a micrograph of CBS under that microscope, and you can see these long, sort of thin string coil-like shapes. And these are even more evident if I show you a zoomed in portion of this micrograph. You can see it zigzagging up and down all over the place, and they're all various different lengths. And with this information, I was able to determine multiple structures of full-length CBS, both in the basal and activated state. And in both these structures, we can see that CBS forms these long oligomers that consist of the dimeric unit found in the crystal structure, but they're interacting at the regulatory domain. And if you want to find out more information on this and other cell biology that we've done with our collaborators, this work was recently published in Nature Communications in April. So in summary, in terms of our fragment-based screening, we have attempted multiple campaigns with a crystal system. However, we were hindered by poor data. We are currently optimizing two constructs of CBS for future campaigns, and they are looking far more promising. Importantly, the HCU network funding has allowed us to gain an industry partner who is funding in our RA, Xiaoju Tang, who's done the majority of the crystallographic-based work which I talked about today. And he continues to work on the fragment-based screening and of the characterization of CBS binders. Our work with the industry partner has also resulted in discovering a novel metabolite that binds to CBS, and this is an area which we're actively investigating. And additionally, we have used cryo-EM to determine the full-length structure of CBS, showing that it forms these oligomers, and we want to understand what role, if any, this oligomerization plays in classical homocystinuria. So I just want to thank Wyatt for supporting me in this work. I really want to thank Xiaoju Tang for doing a lot of the hard work on the crystallographic side base of things for CBS. And I wish to thank everybody else in the ULab. I especially want to thank Arnaud Basile for his assistance to Xiaoju in terms of the crystallization of CBS. And finally, I just want to thank Pfizer, the Wellcome Trust, and the Structural Genomics Consortium, who initially funded this work 10 years ago. And I also wish to thank the HCU network for funding this work. And thank you all for listening.